Bismillahirrahmanirrahim. Topic: pancreas. It is an elongated organ, light tan or pink color. Tan mean yellowish brown. Location: it is present in close proximity to the duodenum. Proximity mean close or near. Morphology of the pancreas consists of three regions or parts. First is known as the head, then body and tail. The head region is present closely to the duodenum. This is the gallbladder and this is the bile duct. With the help of bile duct from the gallbladder, the bile comes into the duodenum and for the process of digestion. Head is an expanded portion, lies in the C-shaped region of the duodenum, to which it is intimately or closely attached by means of connective tissues. Both the pancreas and duodenum, they are connected by a common blood supply. The body and the tail, they extend across the midline of body toward the hilum. Hilum mean depression of the spleen. Spleen is a lymphatic organ. This is the hilum or depression of the spleen. These purple color, these are actually the pancreatic islets known as islets of Langerhans. Consist of different types of cell including alpha cell and beta cell. Islet of Langerhans, they are endocrine in their nature, means they release hormones including insulin and glucagon. This is the bile duct with the help of which biles from the gallbladder enters into the duodenum. This is duodenum, the main pancreatic duct, and these yellow color cells, these are known as the acinar cells. They are exocrine in their nature, secrete digestive enzymes. The islet of Langerhans, they are surrounded by means of acinus. What is acinus? A group of acinar cell is known as acinus. The main pancreatic duct, that is the main pancreatic duct, also known as duct of Wirsung, in the honor of the scientist Johann Georg Wirsung, that extend to the duodenum. Pancreatic juice from this duct and bile from this bile duct empties into the duodenum via or through ampulla of Vater. This opening, the Vater, it is named after the Abraham Vater, while ampulla means cavity or canal. At the end or opening of this ampulla of Vater is provided by means of sphincter of Oddi, named after the scientist Ruggiero Oddi. Sphincter of Oddi, it is actually the valve that controls the flow of bile and pancreatic juice into the duodenum. Structure of pancreas: have exocrine cell known as acinar cell or acini. This single cell, this is known as the acinar cell, while the group of acinar cell or the plural is known as acini, and their associated ducts. Within the exocrine tissue are cluster of this islet of Langerhans, named after the scientist Paul Langerhans. They are actually the group of cells look like an island in the pancreas. These are endocrine in their nature, release the hormone insulin, glucagon and some other hormones. The cells of the islet of Langerhans or the endocrine part: alpha cell, beta cell, delta cell and pancreatic polypeptide. The acinar cell and duct, they form 80 to 90% of the pancreas. Islet of Langerhans make 2%, while the connective tissues, they form 8% of the pancreas. This is a single acinar cell. 20 to 40 acinar cell combine together to form a unit known as acinus, and these are the centroacinar cell that is located within the acinus, and this is the intercalated duct. The content of acinus, that is the digestive enzymes, from this acinus they will move toward the intercalated duct. From the intercalated duct, the content will move to the interlobular duct. From the interlobular duct, it moves to the main pancreatic duct, and from the main pancreatic duct, it will move to the small intestine. Function of acinar cell, it secretes the digestive enzymes. Centroacinar cells, along with the interlobular duct, they release the fluid and electrolyte. Islet of Langerhans consists of different cells. For example, alpha cell, the function of which is to secrete glucagon. Beta cell secrete insulin. Delta or D cell secrete somatostatin, the function of which is it inhibit insulin and glucagon secretion, means oversecretion. Once the function is done and no more insulin and glucagon is required by the body, then their secretion will become inhibited by the D cell. F cell, F because it is a family of peptide, also known as pancreatic polypeptide. It secretes pancreatic polypeptide, the function of which is inhibit gallbladder contraction and pancreatic exocrine secretion. When the body do not require more bile and enzymes from the pancreas, their secretion will become inhibited. 500 to 800 ml of pancreatic juice or fluid secreted per day. Characteristic, it is alkaline, colorless, odorless, means don't have any smell. Acinar cells endoplasmic reticulum secrete isozyme. It is actually the collection of the enzymes. Some of the enzymes as secreted as zymogen, packed in zymogen granules. What is zymogen granules? Specialized storage organelles in pancreas that allow the sorting, packaging and secretion of the digestive enzymes. Zymogen, it is the inactive form of an enzyme. It will become activated when reached to the duodenum. Enzymes released by the pancreas, amylase, it is present in already active form. It function optimally at pH 7. The function of amylase, hydrolyze the starch and glycogen to glucose, maltotriose, that is composed of 3 glucose unit, and dextrin, that is the polymer of D-glucose. Second, lipase, that work or function optimally at pH 7 to 9. The function of lipase, emulsify and hydrolyze fats in presence of bile. Third one, protease, that is responsible for the protein digestion. It is secreted as proenzyme. Pro word means before, means this enzyme is secreted in inactive form and requires the activation for the proteolytic activity. Proteolytic means protein lysis. Duodenal enzyme enterokinase convert the trypsinogen to the trypsin. The enzyme that is released in inactive form from the pancreas, it is trypsinogen. When it reaches to the duodenum, in the presence of duodenal enzyme enterokinase, it will become activated into trypsin, the function of which is protein digestion. The trypsin in turn activate other enzymes. First one is the chymotrypsin. It cleaves the peptide chain at points containing tyrosine, phenylalanine or tryptophan. Means this enzyme breaks down the polypeptide chain at the specific position where these amino acids are present: tyrosine, phenylalanine and tryptophan. Second, elastase, the function of which is break down elastin. Elastin is a protein that is present in the connective tissue. Third one is the carboxypeptidase. It hydrolyzes or cleaves a peptide bond at the carboxy terminal. Terminal word is for the end. Carboxy terminal mean this is the structure of the amino acid alpha carbon, to which 4 different groups are attached: hydrogen, amino, alkyl and carboxyl. In a polypeptide chain, when cleavage occurs at this carboxyl end, that is performed by the enzyme carboxypeptidase. Phospholipase helps in the phospholipid splitting, and trypsin. Along with the activation of these enzymes, trypsin also hydrolyzes bonds by cleaving peptide on the C-terminal end of the amino acid. C-terminal end means where carboxylic acid group is present. But when specific amino acids are present in a polypeptide chain like lysine and arginine. Relation of bile and pancreatic juice with secretin hormone. Secretin, it is the first hormone to be discovered. It is released in the small intestine in response to acid coming from the stomach along with the chyme or liquefied ingesta. Ingesta means something ingested as food or swallowed. Function. The function of secretin is it stimulates the pancreas to release flood of bicarbonate. The function of bicarbonate is it neutralizes the acid that is present in the chyme coming from the stomach. As bicarbonate is a base, thus it prevents acid burns. Established pH conducive to action of other enzyme. Conducive means helpful. Means when bicarbonate neutralizes the acid coming from the stomach, the pH thus develop. It is helpful for the action of other enzyme because only those enzymes can act in the small intestine that work optimally at basic pH. A similar but quantitatively less important response to secretin is elicited or evoked or bring out by the bile duct cells, resulting in additional bicarbonate ions being dumped into the small intestine. Means secretin initially stimulates the pancreas to release the flood of bicarbonate, but in little amount it also stimulates the bile duct cell to secrete bicarbonate ion, the function of which is to neutralize acid coming from the stomach along with the chyme. Exercise. Which of the following converts pepsinogen to the active form of pepsin in the stomach? The answer is HCl. Second, which of the following does not manufacture digestive juices? Answer, kidneys. Third, which of the following is not a function of the liver? Answer is manufacturing insulin. Fourth, which of the following is not a part of accessory organ in the digestive system? Answer, large intestine. Fifth, what is the function of soft palate? Answer is option number C, to keep opening to nasal cavity closed during swallowing. Sixth, an enzyme that acts only in acidic medium is pepsin. Absorption is maximum in the small intestine because of presence of villi. Next, appendix is the part of. Answer is cecum. 9. Bile juice is alkaline. 10. The digestive juice that is almost neutral is saliva. 11th, which of the following is not a function of the small intestine? Option D, transport the residue to the anal canal. 12. Which of the following statement is false regarding the function of large intestine? Option D, digestive activity remain high in the large intestine.